Hello everybody, my name is Bruno Folchi, AgroVenture Director. Today we will talk about Easy Yeast, our biocontrol agent based on Papiliotrema terrestris strain PT22AV. In this video we will give you a quick reminder about how Easy Yeast works on soil-borne diseases. And then we will anticipate a study from University of Molise in Italy, soon to be published, with their findings about Easy Yeast soil ecology and volatilome activity and production. We will then finish with a proposal to the IR4 team to consider studies to evaluate potential synergies between our yeast biocontrol and agro-industrial organic waste management. We introduced Papiliotrema terrestris during the last industry technology session. Here a link to the presentation. We mentioned its interesting soil-borne disease control for nematodes and pathogens, cited by biostimulation, salt stress mitigation, and general contribution to soil fertility and plant health. We have an extensive set of field trials confirming Easy Yeast biocontrol of nematodes such as meloidogene species. 21 trials on tomato strawberry show comparable control to biological and chemical standards. Here you have 8 trials on tomato with Easy Yeast performing similarly to fluopiram and Paecilomyces lilacinus and showing quite good potentials in integrated pest management with chemicals. Performance on fungal pathogens is similarly good. We have numerous trials on Pythium, Fusarium, Rhizoctonia, tomato, potato, strawberry and lettuce, again with comparable results versus biological and chemical standards. Here 6 trials on tomato where you can see Easy Yeast performance compared to Trichoderma species mixes and standard chemical reference propamocarb phosetyl mixes. Let's talk now about Easy Yeast mode of action. We know Papiliotrema is an efficient plant surface colonizer, therefore has strong space competition added to other mode of actions like induced systemic resistance, biostimulation, etc. But we could not explain how it controls nematodes, in particular how juvenile stages of meloidogene are controlled to levels comparable to phostiazide and abamectin. And an answer to this question is most likely coming from the new studies on soil ecology and how they determine the production of a set of active volatiles. This graph shows Papiliotrema terrestris population dynamics after a single application in 2 different agricultural soils. Population decline vary by soil, but stability is reached at several logs higher level versus natural population background already at around 100 days from application. We concluded that Papiliotrema terrestris therefore becomes a dominant, persistent and beneficial soil colonizer. Metagenomic studies confirm Papiliotrema terrestris genus increases significantly in both inoculated soils, while causing significant decline of pathogenic populations. After confirming Papiliotrema soil colonization and persistence, we measured its ability to grow when exposed to root exudates. And we confirmed that the rhizosphere is a very favorable environment for its growth, considering its ability to colonize plant surfaces and roots in particular. It is in fact at root level that Papiliotrema can fully express its mode of actions. In order to identify additional mode of actions for Papiliotrema terrestris, we ran a test to identify its antifungal activity against 5 different pathogen species: Fusarium, Rhizotonia, Sclerotinia, and Alternaria. In dual culture, we confirmed that Papiliotrema doesn't produce water diffusible antifungal compounds unlike Bacillus and Trichoderma, while the dual plate gave quite interesting results. In this test, pathogen interacted with Papiliotrema only via its volatile compounds, thus simulating what happens on soil with low air mobility. And the test, as you can visualize, confirms that Papiliotrema volatile compounds inhibits significantly pathogen mycelia growth and colony diameter, as you can see from this graph, where inhibition ranges from 40% in Fusarium, 80% Rhizotonia, 70% Sclerotinia, 50% for Alternaria. Papiliotrema terrestris volatilum also inhibits reproduction and survival structures such as conidia and sclerotia, as you can see from this graph, showing fast decline of Fusarium conidia germination, as well as mycelium surface. You can see it in the picture. And same for sclerotia, decline in germination as well as colony diameter. We determined the volatiloma composition and the minimum inhibition concentration of each component versus 3 representative pathogens, Fusarium, Rhizotonia, and Sclerotinia. And we identified the most active components with ethanoic acids and other branched chain alcohols. Alone they work, but they work even better together in a bouquet. The volatilome also works very well, is quite active with melodogene species, explaining why Papiliotrema terrestris has such a good nematostatic activity. We therefore could conclude that Papiliotrema volatilome act as biofumigants and are a relevant component of its mode of action. Now, how can we maximize Papiliotrema terrestris volatiles production and therefore its biocontrol activity? And the answer is by increasing availability of the volatiles precursors, which are a set of branched amino acids, which are very abundant in root exudate, microbial biomass, we know that, but they are extremely abundant in agro-industrial waste, such as animal waste like manure, decomposition plant residues, and food organic waste. We can therefore conclude that volatiles are a relevant part of Papiliotrema terrestris mode of action on soil, and that their production is linked to the presence of organic matter on soil. And therefore, our research collaboration proposal to the IR4 team is about investigating 2 different areas. First of all, what kind of agricultural industrial organic matter can maximize Papiliotrema production and biocontrol activity, and at the same time, can Papiliotrema terrestris inoculation to organic waste improve their degradation and stable soil incorporation? Thanks for your attention.